I want to correct my introduction. I'm not emeritus yet, although I may look like it. I want to thank the German Cryonics Society for inviting me and also for tolerating my forceful change in the agenda so that I could speak earlier than I was set to speak. I took a long time in thinking of what I would talk about to the Cryonics Society, and I came up with the idea that my work on transplantation in the retina has some relevance to cryonics because it attempts to repair a part of the central nervous system. And most of the candidates we'll freeze and put into storage will be elderly people, and if you ever revive them, you'll have to do a lot of repair. So repair of the nervous system is an interesting topic that's worthwhile to present here. You're looking at a slice of the monkey retina in the area of the fovea, right up there. And you see it has 3 neural layers. Is there a pointer here that I could use? It has the photoreceptor layer with its nuclei, it has the internuclear layer, and then it has the output layer above, which is very thin. And here it has a structure called the retinal epithelium, which neutralifies these highly specific differentiated photoreceptors. And here you're looking at a complication that afflicts the monkey and man, and that's called age-related macular degeneration. The aging of this epithelial layer, which you're born with and it never changes, it's the same cells you're born with as you die with. And because of that longevity, yes, thank you. Because of that longevity, these cells show tremendous aging effects. And what you see here is a drusenoid complication in the center of the retina that's damaging these epithelial cells, which in turn will damage the photoreceptors. This is a leading cause of blindness in the Western world. People over 55 are mostly sensitive to being blind because of this problem, age-related macular degeneration. And the root cause of it is aging. How do we advance this slide here? I can't.
Hit space.
It's what?
Hit the space bar.
Thank you. This is a 1-year-old section from the epithelium of a monkey. And you see it has mitochondria, has melanin, and endoplasmic reticulum, which is fairly clear. If you look at a 35-year-old section, you'll see this enormous collection of what is essentially garbage material within this cell, due to reasons we still don't understand. But you see a tremendous difference between this and the previous slide with elongated mitochondria. Elongation of mitochondria frequently indicates stress to the cell. And one of the major causes, we think, of aging is due to oxidative stress. And these elongated mitochondria are a clue to that process. You can take these epithelial cells and culture them. And here you see pigmented, and these are human retinal epithelial cells in a culture plate. And when you put them in culture, they do what they never do in vivo, they divide. And you can see them dividing here. You can actually take a trephine slab of these epithelial cells, fold it, and move it around, and try to, as I tried 20 years ago, to transplant these fresh young cells, replacing the old cells in the same region of the retina. This is an owl monkey retina, which is very unique in the sense that the eyeball has a gelatinous material that makes it difficult to get to the retina. The owl monkey has a fluid, watery vitreous, which you can remove quickly. Here to try to transplant these cultured cells into the retina, I started by removing the whole front of the eye, folding it over, then removing the vitreous fluid and looking at the neural retina, which is the neural part, the 3 layers I showed earlier. I actually removed the neural retina and could look at the epithelial layer here. At first, the attempt was to remove the old cells. And you can see the epithelial layer through the microscope, and with a very small wand, you can wipe off the old cells. And then you can take the cultured cells, dissociate them, and try to put them in the place of the cells you had removed. And in doing that, you have to be able to know that you're transplanting the transplant cells. You have to identify the transplant cells histologically. And this here is an identifier. You place. Yes, you place the radioactive thymidine into the cultured media, and when the cells divide, they pick up the thymidine in their DNA and become markable. You can identify them from the host cells, which is essential in transplantation experiment. And here you see that the little dots here are in the nuclei of these epithelial cells, and they have formed a new epithelial layer doing what we hoped that they would do. Remove the degenerate cells and replace them with markable, healthy young cells here. Of course, to do that, we had to remove the neural retina to be able to see things clearly. And that, of course, means that this is not functioning at all. So over the next years, we tried to develop a way of introducing the transplant cells inside the neural retina without removing it, so we have an intact retina. This shows the pipette we use in the introducing of cells through a little bleb detachment. We damage a little bit the neural retina, but we're able to get the cells into what is called the subretinal space. Here you see a pipette, which is used in the surgery, with the dissociated pigmented epithelial cells. And you first produce a hole in the neural retina, and here's the layer we want to correct. And then you can inject either a patch of epithelial cells, or you can inject dissociated cells. This led to a very interesting experiment which got a lot of attention to transplantation of epithelial cells. This is a Royal College of Surgeons rat, which has a defect in the epithelial layer. These cells here are defective. They fail to eat up, phagocytize the growing photoreceptor outer segments. Your visual receptive cells are like your fingernails. They're growing every day, and about 10% is removed every day by the epithelium, which probably is responsible for the buildup of the garbage material. Anyhow, if you don't do anything to the Royal College of Surgeons strain of rats, the defect in the epithelial cell leads to degeneration of the photoreceptors and blindness within 1 month. But if you transplant healthy epithelial cells, you can stop the degeneration. This, I contend, is the first example of transplantation stopping a degeneration in the central nervous system. There is no other. There's another example of it now in a mouse model where the same thing can be shown. This is another example of the transplanted epithelial cells, which are pigmented in an albino rat. And you see the survival of photoreceptors, which would be dead if these cells were not nourishing or helping them nutrientally. We went on to do this in monkeys in anticipation of trying to bring the technique to humans. And here is a rhesus monkey where we transplanted cells here and transplanted cells into the fovea. We have difficulty in the foveal area, which is the area of high-resolution vision and the most important part of the retina. Almost 90% of your visual brain collects from this area here. but it's more difficult to perform the surgery. Here's an example of the monkey retina with transplantation, and notice we have not been able to remove the epithelial layer here, but we can get the retina attached to the transplanted epithelium. Doesn't look good, but it's. Here's another example of the transplanted pigmented cells here in the monkey over the host epithelial cells. It's not what we'd like. We'd like to remove this and then put this in that position. But still the photoreceptors survive for 6 months after transplantation, nutrified by these transplanted epithelial cells. When this work was published, I was invited by the Karolinska Institute to try to attempt to use it in a type of complication of age-related macular degeneration. In about 10%, or maybe a little less, of patients with this age-related macular disease, they develop vessel growth coming from behind the epithelial cells, and this vessel growth goes into the neural retina and leaks and causes destruction. So that and it completely destroys the epithelial layer. So it was thought if we could transplant epithelial cells into the retina after removing the blood vessels, we might restore vision. This is the first human transplant done in Sweden. And you see the transplanted area over an area that had its blood vessels removed. This looked good initially. You see, here's the transplanted one patient. Here it is being tested with a little light flash that could be put anywhere in the retina. And you ask the patient, does he see or does he not see the flash? And you see here, he's using this area where the transplant is, not this area here. The light spots indicate he's seeing light. Here at 1 month later, he's still seeing light. I don't know if you can see this over this computer face. He's still seeing light over the transplanted area at 1 month. But at 3 months, look what's happened. The ability to see over the transplanted area has gone. And he's now looking with his paraphobia, with much poorer resolution. So for 1 month, we had success, but for 3 months, we didn't. That was a disappointment. If that had worked, I would be more famous than I am now. One of the thoughts was that it's due to rejection, because we're putting embryonic human cells into another human being. So it's an allograft. And I contend that it possibly is rejection, but it doesn't rejection doesn't occur across the whole retina. Here is a transplant here in a patient. We were trying to correct this area here, but after surgery, the transplant drifted away. And 3 years later, it's still there and not rejected. But this is a major problem, is rejection. Now, you saw in the previous work and in the work in humans, the epithelial layer was removed by the neovascularization. But in normal people, if you try to transplant cells, you have to remove the original epithelial layer. And it's very difficult to do that through the neural retina. So a technique is now developed where you cut the neural retina at 180 degrees in the periphery, where you're not seeing it all, far out here, cutting the retina there, and then folding it over the optic nerve, allowing you to see the epithelial layer directly, as was done earlier, 20 years earlier in the owl monkey. Being able to see and work with 2 hands on this area is of great value for improving the surgery. And here is an example of an attempt to use this technique, rolling off the retina, the neural retina, removing epithelial cells from the periphery, and putting them over the transplant. This is a homograft, so there should be no rejection. And this is a successful surgical attempt done by a group in Verona, Italy, which shows that you can now get to removing the host epithelium directly and put the transplant over it. The only problem is here, taking these epithelial cells and putting them over here is you're working with old cells full of the garbage. One of the breakthroughs now is stem cell research, where you can actually take a skin cell from the skin of a patient, transform it into a pluripotential cell, and then have it differentiate into an epithelial cell. This means you can obtain a homograft from the same patient and avert rejection completely. So the field is slowly developing, and transplanting of epithelial cells is going on. And it's an attempt to rebuild and repair senile old cells using surgical techniques and modern methods of surgery. This simply shows the concept of removing the senescent cells, and the neural retina has been lifted off so you can see this area directly. Transplanting embryonic cells to this area, letting the retina float over the epithelial layer which has been restored. You're putting new cells into an old retina in exactly the place they belong. And that's not an easy thing to do anywhere else. Well, because of the success, relative success, with epithelial transplantation, a lot of us and other members, particularly in England, have been thinking of photoreceptor transplantation, which is even more exciting, but very difficult. This is an attempt by us in the RD mouse, a mouse that loses its photoreceptors because of the defect in the photoreceptor itself, to transplant embryonic mouse photoreceptors in a microaggregate. And you see it here transplanted into the subretinal space and with outer segments. Here, there are no outer segments because there's been no transplant. So you can actually transplant photoreceptors, put them in the right position with their outer segments touching this epithelial layer, which we talked about before, facing the second-order neurons, hoping that would make communication. To do that would bring vision back to a blind eye. The epithelial cells are neutrophils. They help the photoreceptors live, but the photoreceptors themselves are the cell that responds to light. And putting those photoreceptors in, if they could be made to contact second-order neurons, would bring vision to a blind eye. This is another example, and it shows that if you transplant them with the right polarity, with the outer segments facing the epithelial layer, they survive well. If you have them transplanted in the opposite direction, with the photoreceptors facing the neural retina, they don't survive as well. So you can transplant photoreceptors. The question is, can you make them talk to second-order neurons? And that's the big challenge in this field. Here's an example of electron microscopy. Again, here these little dots mark the transplants. They show that they're cells we've removed from one mouse and put into another mouse using a viral vector marker for the transplants, which is, again, essential. Here is the only indication I've found for possible communication between the transplant, which have been labeled by these LAC-C particles here. This labels this as a transplanted photoreceptor. And here is a synaptic zone. And you see we have labeled not only the photoreceptors we transplanted, but this mouse has labeled second-order neurons with LAC-C. And here we see LAC-C in the second-order neural compartment of the synapse, and LAC-C components in the photoreceptor cell itself, suggesting that there is synaptic contacts, but they're very, very rare. We can, of course, use very nice techniques to isolate photoreceptors. This is an example of an adult human retina obtained from an embryo. We work mostly with 20-week-old human embryos taken from abortion, which has some problems, but they're absolutely beautiful cells. And we use the excimer laser to remove the other cells of the retina to end up with pure photoreceptors here on the epithelium. This is the retina completely. This is after the excimer laser removes the inner parts of the retina. And this is where it's incomplete. The excimer laser was not able to remove all of these second-order neurons, but here you see it, removable. And these are cone photoreceptors in culture having their second-order neurons removed by the excimer laser. So we're able to have foveal cones living in a dish, and we're hoping to put them into the neural retina where there are no photoreceptors. Will they communicate by synaptic formation is still a major problem. In fact, it's one of the major problems in transplanting neurons in the central nervous system. Can you encourage them to form synaptic contacts with second-order neurons? That's a Nobel Prize piece of work. It can be done. Well, here is just a little bit of the history of this in my publications, and some of my publications in the field, showing that you can transplant photoreceptors and have them survive and develop in foreign retinas. What we have to learn is how to make these cells form synapses with second-order neurons. I hope that will be done in our lifetime. But it shows you can repair this part of the central nervous system, the retina, which is part of the brain and one of the most highly metabolizing structures in our body. It's possible to repair it and bring vision to blind eyes in ways that we never dreamed of 40 or 50 years ago. Thank you very much.
Your transplant cells are heavily pigmented. How do you harvest it? As its culture cells?
When the cells divide in culture, they lose their melanin. It's very difficult to get melanin synthesis in these cells. But in a way, they not only lose their melanin, but they lose their garbage material. So if you take old cells, culture them, have them divide, they slightly rejuvenate themselves by losing their garbage material, which we call lipofuscin. And they also lose their pigment. Although there are techniques now that will encourage pigmentation of cultured human or any animal cells, retinal epithelial cells. I think you can get them to pigment themselves, but the ones I worked with here have lost their pigment.
I don't know if I got it all right. Do the transplanted cells develop pigments secondary after transplant?
You saw some here with pigment in them. Some of the transplants I showed you in the monkey had pigmented cells with photoreceptors on top of it. So they all don't lose that pigment. There is pigmentation there.
Yes.
I'm not sure pigmentation is essential because albino people have not good vision, but they have vision. So you don't need the pigment to see or to even help very much in seeing.
Is it known why retina epithelial cells divide in vitro but not in vivo?
That's a very good question. There must be contact inhibition. Somehow, and even the transplants, you would think you might have a tumor. When we put these cells, which were dividing in culture, back into the retina, why don't they start dividing and build a tumor rather than having a small transplant? It's either in the environment, mitosis is counteracted, either in the dish or in vivo. But that's an interesting question. Why putting them in a culture plate makes them start dividing? Otherwise, they never divide during our lifetime, unless you have some damage, some inflammatory or some traumatic experience that damages the cells. They could divide, but not very much.
May there be some ways to establish the relevant conditions in vivo too, maybe by medication?
To stop, but they don't divide in vivo. They're not dividing in vivo.
Yeah, they don't.
They don't divide in vivo.
But you agree they divide in vitro because of certain conditions.
Yes, there must be.
Yeah, but
Yeah, we could perhaps find some technique to stop them from dividing in vitro? Is that what you're asking?
I think he's suggesting that if you could make them divide in vivo, then you might have a repair strategy in situ.
I wouldn't like them to divide in vivo. I would think we might have a tumor.
okay. Yeah, my idea was to avoid the surgery in the first place.
How do you avoid the surgery?
Yeah, that was my idea. If you can identify the relevant conditions that make it happen in vitro, maybe you can trigger or establish these conditions also in vivo by medication and to avoid the surgery in the first place. That was my idea, but maybe it was naive.
Yes, I see. You want to have medication or have these cells divide in vivo without doing surgery.
Yeah.
Yes,
Right.
but that's not so easy.
Yeah.
It's a nice idea.
Yeah. I imagine.
So I'm a little confused about something. Your main challenge at the moment, as I understand it, is that when you transplant photoreceptor cells, they are very bad at forming connections with neurons.
Yes.
Right. But the cells that are really suffering during normal age-related macular degeneration are the RPE cells.
Yes,
If you could just replace the RPE cells,
that's what we're trying to do,
right,
of course.
and leave the photoreceptor cells alone, then the photoreceptor cells would still have their existing connection to the neurons.
Yeah, that would require. That's a good point. That would require doing a surgery before the photoreceptors degenerate. So we have to have this technique so good that we would take a chance to prevent photoreceptor degeneration by transplanting an eye that's still seeing with that macula.
So if you did that after the photoreceptor cells had degenerated,
Then we're in trouble.
Wait. So would it be? I mean, you said that the RPE cells do not divide at all in vivo.
Yes.
But is there any replacement of photoreceptor cells in the context of a rejuvenated RPE layer?
That's what I think we're suggesting with the transplants of photoreceptors. We can certainly transplant photoreceptors over transplanted retinal epithelium. The big problem is the second-order neuron contacts.
Right.
What happens, too, is the glial cells, as a lady here is working on glial cells and photoreceptor vision. The glial cells grow down and appear to block the accessibility of the photoreceptor terminal to get through. And that's a nasty complication. That's why I have a tough time finding any synaptic interaction